Good morning, everyone, and thank you for being here today. I'm Dr. S.K. Singh, and I'm excited to present to you on the topic of cleanroom requirements. Today, we'll be exploring the different grades of cleanrooms, specifically grade A, B, C, and D facilities. Over the next few slides, we'll cover what each grade entails, their importance, and the key standards involved. So let's get started and dive into the world of cleanroom requirements. Let's talk about the regulatory bodies that have been established to ensure the quality of the products we manufacture. These organizations set strict regulations for drug manufacturing facilities to maintain high standards. One of the key frameworks they created is the Good Manufacturing Practices, or GMP, specifically for sterile medicinal products. GMP ensures that the drugs are safe and that they contain the right ingredients in the correct amounts. It also aims to minimize the risks of contamination from microbiological sources, particles, and pyrogens. Now, it's important to understand that cleanrooms don't completely eliminate contamination. Rather, they control it to a tolerable level. In situations where manufacturing processes could introduce defects, a higher level of cleanliness is necessary to prevent contamination. Under GMP, the production of sterile medicinal products is categorized into different grades, A, B, C, and D, based on the environmental control needed. In pharmaceutical manufacturing, cleanrooms are classified by these grades to match the required level of environmental control, ensuring that the final product remains safe and effective. Finally, a well-designed cleanroom system is crucial. This includes validated HVAC systems, pressure differentials, airflow patterns, cleaning procedures, and ongoing environmental monitoring. All these elements work together to maintain GMP compliance and, most importantly, to protect patient safety. Let's take a closer look at the different grades of cleanrooms and their specific roles. Starting with Grade A, this is the highest level of cleanliness we need for critical aseptic operations. Think about tasks like filling, open vials, or sterile connections. These are operations where even the tiniest contamination can cause big issues. To achieve this, we typically use LAF or isolators, which provide a controlled environment with continuous monitoring of both viable and non-viable particles, ensuring the highest standards of sterility. Moving on to Grade B, this acts as the background environment for Grade A zones during aseptic preparation and filling. It's still very controlled, but slightly less critical. The focus here is on strict control of particulates, microbiological limits, and personnel practices to keep contamination risk as low as possible. Then we have Grade C, which is used for less critical stages like preparing solutions or handling components before sterilization. The environment here is controlled, but doesn't require the same level of strictness as Grade B or A. It has defined limits for airborne particles and microbial presence, ensuring safety without the need for the highest level of sterility. Finally, Grade D is our lowest classification. It's suitable for initial or less critical operations, such as washing components or preparing materials before they move into more critical stages. It provides a clean, controlled environment, but doesn't require the stringent controls seen in the higher grades. Understanding these classifications helps us ensure that each step of the process is performed in the appropriate environment, maintaining product integrity and patient safety throughout. Now, let's discuss the key aspects of maintaining aseptic conditions during the filling process. First, the assembly of filling equipment must be done aseptically, ensuring that all connections made after the final sterilizing filter are also sterile. Whenever possible, these connections should be sterilized using steam-in-place methods to maintain integrity. Aseptic compounding and mixing are critical steps, as they help prevent contamination before the product is filled. Replenishing sterile bulk products, containers, and closures needs to be carefully controlled to avoid compromising sterility. When removing items from sterilizers, unprotected items, like those without packaging, must be cooled and handled properly to prevent contamination. Staging and conveying sterile primary packaging components in the filling line is another important point. These components should be kept unwrapped only in controlled environments. During filling, sealing containers such as ampules or vials, and transferring open or partially stoppered vials must all occur under aseptic conditions. Loading the lyophilizer also needs to be done carefully to prevent contamination. It's worth noting that sinks and drains are prohibited in Grade A areas to avoid contamination risks. High-risk operations, such as filling zones, stopper bowls, open ampules, vials, and making aseptic connections, require extra vigilance. To achieve Grade A cleanliness, a laminar airflow cabinet can be used within a Grade B background. This setup can provide an environment equivalent to an ISO 5 cleanroom, both at rest and during operation. Overall, maintaining strict aseptic practices at each of these steps ensures the integrity and safety of the final sterile product. The Grade A area is specifically dedicated to high-risk operations. This includes critical processes like fill and finish, working in the filling zone, stopper bowls, open ampules, vials, and making aseptic connections. These activities require the highest level of cleanliness and control. A CGMP Grade A environment is considered equivalent to an ISO 5 classification, both when equipment and personnel are at rest and during operation. This means that regardless of whether staff are actively working or not, the cleanliness inside the cleanroom must consistently meet Grade A standards. To maintain this level of cleanliness, we use specialized equipment like laminar flow cabinets or laminar airflow hoods, known as LAF. Even if the surrounding air is only classified as Grade B, a laminar flow cabinet can create a localized environment that achieves Grade A conditions. The key to reaching Grade A cleanliness is controlling airborne particles. As outlined in the table, the number of airborne particles allowed in a Grade A environment is very low, ensuring a sterile and safe space for sensitive operations. Maintaining this environment is crucial for product quality and patient safety. Now, let's talk about the background support for Grade A environments when they are not housed within an isolator. This support is essential for maintaining the integrity of the Grade A space, especially during activities like conveying or staging equipment and components. While doing this, it's important that these items are protected from the surrounding environment to prevent contamination. Moving on, I want to highlight that sinks and drains are actually prohibited in Grade B areas. This is because they pose a risk of contamination, so their absence helps maintain the cleanliness standards required. Regarding air quality, Grade A environments are maintained at ISO 5 when at rest, and they remain at ISO 7 during operation. This difference ensures that the environment stays as clean as possible, whether it's idle or actively being used for aseptic work. Another critical point is the particle monitoring system. This system must be in place with alarms that activate if particle limits are exceeded. It provides real-time monitoring and helps ensure ongoing compliance with cleanliness standards. The background zone for Grade A is also an important concept. It acts as a controlled environment that supports aseptic preparation and filling processes, helping to minimize contamination risks. Finally, I want to clarify that clean rooms designated as Grade B are used specifically for aseptic preparation and filling, making them a crucial part of the overall sterile manufacturing process. This slide highlights how each element works together to uphold the stringent standards necessary for pharmaceutical and biotechnological production. Let's talk about sinks and drains first. In Grade A and B areas, sinks and drains are not permitted, as they can be sources of contamination. Moving on to the Grade B room in your GMP facility, it's essential that it's equipped with a particle monitoring system. This system should include an alarm that triggers if the particle limits are exceeded, helping us maintain the required cleanliness standards. Remember, the Grade B cleanroom environment is used for aseptic preparation and filling, so maintaining control over airborne particles is critical. When the room is in operation, it functions at an ISO 7 level, which means it has a certain allowable particle count. But when it's at rest, that is, not in use, it needs to meet even stricter standards, equivalent to an ISO 5 environment. Specifically, at rest, the maximum allowable particles of 0.5 micrometers are 3,520 per cubic meter. During operation, this limit is naturally higher, up to 352,000 particles per cubic meter. This distinction highlights how particle counts can fluctuate depending on whether the room is actively being used or not, but overall, strict controls are essential to ensure product safety and quality. This all ties back to our overall goal: maintaining a controlled environment that ensures the integrity of our aseptic processes. Let's move on to slide 8. Here, we focus on the preparation of solutions to be filtered. This includes sampling and dispensing, which are critical steps to ensure the integrity of the final product. Even though some operations are less critical, it's important to handle all steps with care to maintain quality. We operate under ISO 7 standards when the clean room is at rest, and ISO 8 standards during operation. Monitoring is a key aspect, and it really depends on our quality risk management approach. This means we tailor our monitoring efforts based on the potential risks involved. Finally, this slide highlights the use of these clean rooms for filling products that will undergo terminal sterilization, especially in a Grade C environment. Maintaining proper conditions in a Grade C clean room is essential to ensure product safety and compliance throughout the process. Let's talk about monitoring the Grade C area. It's important that this is done in accordance with quality risk management principles. This means using a systematic approach to assess, control, communicate, and review risks to the quality of the medicinal product throughout its entire life cycle. Now, Grade C cleanroom spaces are designated for performing steps in sterile product manufacturing that require less stringent environmental controls. To give you a sense of the cleanliness standards, the airborne particle classification for Grade C when at rest is ISO 7, and when in operation, it's ISO 8. At rest, the allowable number of particles per cubic meter is 352,000. When the room is in operation, that limit increases to 3,520,000 particles per cubic meter. Understanding these classifications helps us maintain the appropriate environment for manufacturing, ensuring product quality and patient safety. This connects to our overall theme of maintaining strict controls and continuous monitoring throughout the production process. Now, let's talk about the key steps in maintaining a clean and controlled environment. First, we focus on the cleaning of equipment, which is absolutely critical to prevent contamination. Once equipment, components, and accessories are cleaned, handling them properly after cleaning is essential to keep their sterile state intact. Next, before sterilization, we assemble cleaned components, equipment, and accessories under HEPA filtered airflow. This step helps ensure that no new contaminants are introduced during assembly. For sealed or closed systems, we use intrinsic sterile connection devices to assemble sterilized stainless steel components, or SUS, maintaining their sterility throughout the process. It's also important to recognize that some operations are less critical. These are typically classified as less sensitive in terms of contamination risk. Now, regarding cleanliness standards, when the room is at rest, it maintains an ISO 8 classification, which indicates a controlled environment with specified particle counts. However, it's important to note that the ISO 8 standard isn't defined for operational conditions, only at rest. Finally, I want to highlight that the clean room environment we're discussing is considered the dirtiest according to GMP guidelines, specifically classified as a Grade D clean room. This underscores why strict procedures and controls are so vital in this environment to ensure product safety and quality. So, just like the Grade C clean room in a GMP facility, the Grade D area is designated for performing less critical steps in the manufacturing of sterile drugs. It's important to understand that the Grade D environment can often serve as a background zone, depending on how your cleanroom is designed and set up. Essentially, this is the least clean area that still meets GMP requirements for sterile products. When it comes to airborne particles, the classification for Grade D at rest is equivalent to an ISO 8 cleanroom. However, Once the area is in operation, the particle levels aren't fixed. They can fluctuate depending on the specific processes happening inside. That's why we often refer to this as a clean non-classified or controlled not classified area. This means that while it's the least critical zone, it still plays an essential role in the overall cleanroom design, ensuring that less critical steps are performed under controlled conditions without compromising the sterility standards required for the final product. Let's take a closer look at the different cleanroom grades and their specific roles. Starting with Grade A, this is the highest level of cleanliness we need for the most critical aseptic operations. Think of it as the cleanroom where sterile filling and aseptic connections happen. We achieve this through unidirectional airflow, often using laminar airflow cabinets or isolators, and we continuously monitor particles and microbiological contamination to maintain the environment's integrity. Moving on to Grade B, this serves as the background environment for Grade A processes. It provides a controlled setting during aseptic preparation and filling. Stringent controls are applied here, including managing airborne particles, monitoring viable microorganisms, maintaining pressure differentials, and controlling personnel movement to prevent contamination. Next, Grade C is used for less critical steps, like solution preparation or handling materials before they go through sterilization. While it still requires environmental controls and monitoring, these are less strict compared to Grades A and B, reflecting the lower risk of contamination in these activities. Finally, Grade D is the most basic, suitable for early-stage or non-critical operations. This includes activities like component washing or material preparation. Here, we focus on maintaining basic cleanliness standards and GMP controls, but the environment doesn't need to be as tightly controlled. In summary, these grades, A, B, C, and D, form a hierarchy of cleanliness, each tailored to the level of risk associated with the specific process step. This structured approach helps us ensure product safety and compliance throughout the manufacturing process. If you need any further support or have questions, please don't hesitate to reach out. We're here to help you succeed. Thank you so much for your time and attention today.